Good morning, students. Now, this much time we were discussing and we were dealing with the chapter 8, that is human health and diseases. Okay, and we have seen many diseases, then the immunity, different types of immunity, and some more diseases we have to discuss in the same chapter. Out of that, today we are going to discuss about the AIDS. I think everyone knows what is the AIDS and what is the full form of AIDS. Now, let's see each and every step that is taking place in this disease, that is in the AIDS. Now, first of all, you can see the picture that is given here. This is the AIDS virus, and we know the name of the AIDS virus is HIV virus. Okay, and here even you can see the different parts that has been given here. One is, you can see here the HIV enzymes are here and their genetic material, that is the RNA, and there is a covering here, and the covering is called as the capsid, and this capsid is made up of a protein. Okay, so I think the structure of this virus, everything we have dealt in the class 11 syllabus. Clear? So this is a picture, overall picture of the AIDS virus. Now let's go to the detailed study of the AIDS. What is the causing organ of the AIDS? So we know that the full form of the AIDS is acquired immunodeficiency syndrome. Now let's deal with this sentence, acquired. Okay, what do you mean by the word acquired? Acquired means something that we are getting during our lifetime. Okay, so that means this is not any congenital disease. And you know the meaning of congenital disease. Congenital disease means something that we get during our birth or at the time of the birth. Okay, that means something we are getting from the before the birth or at the time of the birth. But this disease is an acquired immunodeficiency syndrome. So first part is over, acquired. So this is acquired during our lifetime. And immuno, what do you mean by the word immuno? Immunity. And the next word, third word, is deficiency. So what is deficient when we are suffering from this disease? That is our immunity. Okay. Now the fourth part of this full form, that is called as a syndrome. Now what do you mean by the word syndrome? Syndrome means a group of symptoms. Clear? So it's not a single disease. Yeah, it's not the symptom shown by a single disease. It's a group of symptoms has been shown, and because of these, because of the deficiency in the immunity system, what is happening? A group of symptoms or group of diseases will attack our body. Okay, so that is the word meaning. Yeah, that is the full form of the AIDS, acquired. That means we are getting during our lifetime, and immunodeficiency, together we can spell it out. Immunodeficiency means we are deficient in the immunity, and syndrome means that is a group of the symptoms. Clear? The word is clear. So they are giving the part like this disease is acquired during our lifetime, so it's not any congenital disease. Then what is the name of the virus that is causing this disease? That is human immunodeficiency virus. Clear? So you call it as HIV virus. Again, I'm telling human immunodeficiency virus. Now, what is the genetic material? We have seen that structure in the first page. In that, we have shown that what is the genetic material present in the AIDS virus? That is the RNA. And this group of virus, you will call the name as the retrovirus. Okay. So HIV is a retrovirus, and their genetic material is made up of the RNA. Now, again, we define what is a syndrome. That is means a group of the symptom. Clear? So, HIV, they are a group of retrovirus, and their genetic material is made up of RNA, and they show a group of the symptoms. And the first case of the AIDS has been reported in the year 1981. Clear? So this is an introductory part about the AIDS, how, what is the name of the virus, and what's the word meaning of that AIDS, acquired immunodeficiency syndrome, and what is the genetic material that is present in this virus. Okay. Now the next part we are going to deal is how it is getting transmitted from one person to another. I think you people might be having any idea of how they are getting transmitted. Have anyone has any idea about how this AIDS get transmitted from one person to another? Of course, we know 2-3 points might be like sharing of the needles. Okay, sharing of the needles or the sharing of the blood, or sometimes if the mother is infected with the HIV, it can pass on to the baby but through the placenta. Okay, I think these things we know. And the drug abusers, if they are taking the drugs through injections and all, so the needle used by the one person, if it is used by another person, those who are having HIV, then what will happen? It get transmitted from one person to the another. Okay, and another thing, having multiple partners, with having sex with the multiple partners, that is also one of the main thing that is in the helping in the mode of transmission. It's not helping, that is occurs in the transmission of the AIDS from one person to the another. Okay, so let's see one by one. Sexual contact with the infected person. Clear? Then by the transfusion of the contaminated blood and the blood products. So this is mainly, we have to be very careful while we are accepting the blood from some other person. And I think the blood bank is all having all kinds of precautionary measures. They will check whether the blood collected from any person is there, whether contaminated with this virus. After that only they do the transfusion process. So this is one major thing, that because we should not have any ignorance, and because of our ignorance we should not suffer from the AIDS. Then by sharing the infected needle, especially in the case of the people who are drug abusers, and they are the intravenous drug abusers. Intravenous drug abusers means the where you are using the needle, okay, and it will go up to the vein. So that one you have to be careful. And the fourth one, we can say that if the mother is infected, then what will happen during the time of the delivery and all, the child can be affected through the placenta. Okay, so these are the different modes of transmission. So first one, sexual contact with an infected person, and by transfusion of this contaminated blood, and sharing the needles, and from the infected mother to the child through the placenta. Okay, now another important thing that we have to notice that when the infection starts in our body, nahi to when the HIV infect our body, it's not that they will start producing the symptoms suddenly. No, what will happen? There is a time lag. There is a time lag between the infection and the appearance of the symptoms. Okay, this is the important thing. Like now we are discussing about the coronavirus. We are all having the 14 days time. Why? Because that is the time period by which the virus will start showing the symptoms. The same thing with here, but here the time lag is more. Okay, it can start from few months, or sometimes it will go to even several years. And the several years means sometimes it can go like 5 years, sometimes it can go up to 10 years, like that. So that is one of the drawback we can say, because the after the entry of this virus to our body, we are not able to identify that whether the virus has infected us, because the time lag between the infections and the appearance of symptom is more. Clear? So this thing we have to be careful, because don't think that HIV, if the HIV came to our body and we will start showing, it's not like that. It will take few months to the several. Okay, so these are the mode of the transmission. Now we are going to the replication of that virus. And what is the name of the virus? HIV we are telling, but the group of the virus is called as the retrovirus. I'm showing you the life cycle, yeah, showing you how the infection is starting in a normal healthy person. Okay, now can you see this diagram here? I have given, see, retrovirus has been given here. So you can see here retrovirus. Then what is happening? This retrovirus is having the viral RNA as their genetic material. Okay, they are having the RNA as the genetic material, and you can say that they are having the viral protein code. Now what will happen? This viral RNA code, they will infect a normal cell. So you can see this one, the rectangular shape has been drawn, and this is a normal animal cell, and the boundary that they are showing here is the plasma membrane. Okay, now what's happening here? This virus infect a normal cell, and this structure, that is the viral RNA, and this viral RNA is entering into the animal cell, yeah, host cell. Okay, now what is happening here? This RNA will synthesize a DNA. Okay, so first RNA is here. Now this RNA will be synthesizing the viral DNA. And what is that process called? This process is called as reverse transcriptase. Okay, you have to be reverse transcription. And the enzyme that is used in this process is called as reverse transcriptase. Now I will explain you a little bit later ki what is this process called. So this is the process by which the viral RNA is getting converted into the DNA, and the name of the process is reverse transcription, and we are using the enzyme called the reverse transcriptase. Okay, so now this DNA is here. Now what will happen to this DNA? This DNA is getting incorporated into the host genome. Okay, so this is the host cell has been shown here. So this is the host cell, and along with this host DNA and everything, what will happen? This viral DNA gets incorporated into their genome, host genome. Okay, now after that what will happen? This viral genome that is present here, they will start the infection. Yeah. So when the infection starts, new viral RNA is produced by the infected cell. So once as it has been infected, then after that many more viral RNA has been produced in the infected cell. Now you can see this cell contains more number of infected cell, and they will start reproducing again, and you can see many of the new viruses have been produced. Now what will happen to this new virus? They can infect any other cell. Okay, so here I have written here infected cells are there, they can survive while these viruses are replicated. You can see many viruses have been formed here, many have been formed here, this one, then this one, many viruses have been formed here. So they are replicated, and finally what will happen after their replication? They will be released and they can infect another host organism. So this is the method of replication of the retrovirus. Once again you can see here. So what's happening? The retrovirus is coming here. The retrovirus is here. Now this retrovirus, they have the genetic material of the RNA, and they infect a normal cell. Okay, they can infect a normal cell, and this viral RNA will get into the host organism, and by the process of the reverse transcription, the viral DNA has been synthesized, and the enzyme that we are using here is the reverse transcriptase. Now this viral DNA then gets incorporated into the host cell, and once they get infected, the viruses are replicated, and the number has been increased and they have been released. So we can see here many viruses have been newly produced, and this virus can infect another cell. So infected cell can survive while the viruses are replicated and they are released. So you can draw this figure. It's all present in your NCERT book, and you have to draw this diagram. Sometime this question will come for 5 marks also. So that time you have to draw this diagram. Along with the diagram, all the labeling should be there. Okay, and that is enough for you to score the very good marks. Your full marks you will get. Okay, so you should be familiar with all these terms, and the diagram should be very clear. Along with the diagram, the written part should be there. Okay. Now let's go to the life cycle of the HIV. Already we discussed. Now we will discuss little more detail what's happening in the life cycle of the HIV. Now after getting into the body, what will happen? This virus will enters into the macrophages. Okay, now we know that what is macrophages? They are the special cells of the immune system, and they are responsible for the immunity, and they are responsible for the phagocytosing, and they are responsible for destroying the foreign bodies. So what will happen? This virus will enter and attack the macrophages, or we can call it as helper T cells. Okay, now, already we told that RNA genome, that is the RNA is a genetic material, and they will be converting into the DNA with the help of the enzyme reverse transcriptase. And before that, I will show you the normal mechanism. So here you can see here, this is the DNA. So from DNA, we are synthesizing mRNA, and this process is called as a transcription. This is the general one I am telling. So this process is called as a transcription. Okay. Now, after the mRNA has been made, it is undergoing another process called the translation, and finally the protein synthesis is taking place. So this is the overall step, or you can say central dogma of molecular biology. So what is the central dogma of molecular biology? DNA undergo transcription and they synthesize mRNA, and the mRNA undergoing translation, they synthesize the protein. Okay, this is a normal method. Now what's happening in the case of the retrovirus? RNA is here, so instead of DNA, Their genetic material is RNA. From RNA, the DNA is synthesized. That's why the word has been given, the term has been given as the reverse transcription. So transcription is here. But what happened in this condition? The DNA is the ulta. That means the RNA comes first, then comes the DNA. So that is why it is called as the reverse transcription. I think now it's clear for you, because the normal transcription process, the DNA is transcribed into the mRNA. But in the case of the retrovirus, it's the RNA that is getting transcribed into the DNA, and that's why this process is reverse. So that's why we are calling this process as the reverse transcription. And the enzyme that we are using here is called as the reverse transcriptase. Okay, and finally the synthesis is protein only, but that's why we are calling this step as a reverse transcription, because it's something that has been reversed from the normal process of the transcription, and we need a special enzyme here, and the name of the enzyme is the reverse transcriptase. I think it's clear for everyone why the process is called as a reverse transcriptase. Okay, then let's pass to the next. So we are still discussing about the life cycle of the HIV. Now what happened? Now the macrophages got attacked, and the viral DNA that is getting into the host cell, and they started producing more number of the viral particles. Now what is happening here? The macrophages become the factory of the HIV. Okay, that become the factory of the HIV, because more and more viral cells, viral RNA has been formed and viral particles have been formed. So this macrophages started acting like the factory of the HIV. Okay. Now the viruses that is released from this macrophages, they will start attacking the helper T cell. And you know what is the use of helper T cells? And there is a progressive reduction in the number of helper T cells. There comes the second point, that is immunodeficiency. I think it's clear. It's becoming clear for you. The macrophages became the factory of the virus. Now what's happening here? The virus that is released from the macrophages, they start attacking the T helper cell. Okay, T helper cell, that means the T cells. Clear? Now what's happening there? If the T cells has been attacked, the number of the T cells will become reduced. So there is a overall reduction in the number of helper T cells. Now, if the number of T cells has been reduced, automatically what will happen? Our immunity level will become reduced, and our body will become prone to different types of diseases. Not only a single disease, different types of diseases, sometimes some attacked by the bacteria, virus and fungi, because our body's immunity power has gone down, because again the helper T cells has increased, reduced in their number. The reduction occurred with the helper T cells, and normally our immunity level has become clear. So that's why what will happen, the a person who has been attacked with this HIV started showing like bouts of fever, sometimes diarrhea, sometimes weight loss. These are the symptoms that has been shown by that person. And sometimes what will happen, they start suffering from different infections caused by different microorganisms. Some of the like mycobacterium are the viruses, are the fungi, and sometimes some parasites like toxoplasma, they attack that person. Okay, and that's why again the last part, the syndrome. What do you mean by the word syndrome? A group of symptoms will be shown, because that body has been attacked by lots of microorganisms like mycobacterium, viruses, fungi, and the parasites like the toxoplasma. Clear? And the main symptoms that has been shown that time might be like they're having suffering from the bouts of fever, sometimes diarrhea, and sometimes weight loss. Okay, now the disadvantage is that even though we are showing this symptom like fever, diarrhea, and we don't think that our body contains the HIV, because this is all normal symptoms that some other disease can also share. Okay. So this is about the life cycle of this one, AIDS virus. Now the next one is the diagnose, and the most modern technology that is called as the ELISA. Okay, what's the full form of the ELISA? Enzyme linked immunosorbent assay. So by this method, the diagnosis can be done that whether the person is suffering from the AIDS or not. Now, what's the treatment? Everyone knows that there is no specific treatment for this disease, and there is no medicines available there for the complete curing of this disease. So normally what is the treatment we deal with is that we apply antiviral, antiretroviral drugs. Okay, and it's not completely effective, it's only partially effective. And what's the use of this kind of medicines? It can prolong the life of the patient, but we cannot prevent the death. The death is inevitable. Death has to be there. Okay, but we can have this medicine like antiretroviral drugs so that the little bit boosting of the immunity can be there and the lifespan of that patient can be increased. Understand? So I think it's clear. What is the diagnostic method of the AIDS? The diagnostic method is the ELISA. And what's the treatment? We are treating with some kind of antiretroviral drugs, but it's only partially effective. Okay. Now, prevention of the AIDS. There are some preventive methods that we can follow. The first one, to follow safe blood transfusion. This is a place we have to be very careful, especially the blood banks. Okay, they have to supply the blood with proper checking, with proper labeling. They have to have all kinds of tests, then only they should do the blood transfusion. And the second one, to use the disposable needles. In the previous time, what we used to do, we will take one needle and we will sterilize the needle and we will use on the another person. So now it has been stopped completely. We have to use the disposable needle. And another thing, we already told that from the sexual partners. So we have to, the government is doing this one, like we have to distribute the free condoms to avoid the sexually transmitted mode of the transmission of the AIDS. And the fourth one, to prevent the drug abuses. Okay, so these are the different prevention methods. Now, some organization also involved in dealing with this AIDS. The most important ones are the National AIDS Control Organization, NACO. I think you might have heard about this NACO. Um, and here I have drawn one sign, yeah, logo of the NACO. This is very famous logo, that is National AIDS Control Organization, India's voice against the AIDS. And some NGOs are involved in this one. They are helping out the people and they are helping out the government. And the another third one is the WHO, World Health Organization group. Okay, so these are the main three NGOs. Many NGOs are running, and the most important one we have is the NACO and the WHO. Now, what are the initiative that has been taken by the WHO in preventing this diseases? So you can see I have put the logo for W. This logo is also might be familiar for you people. And what are the different initiative taken by the WHO? You can see they are making the blood from the blood bank safe from HIV. Okay. And the second one, this already we discussed when we discussed about the how we can stop the transmission of this disease, ensuring the use of the disposable needles and syringes in the public as well as in the private hospital. Then controlling the drug abuse, advocating the safe sex, and promoting the regular checkups in the susceptible population. We can't go and have checkups for every person, but if any population is susceptible to this AIDS, then we have to promote the regular checkups in that susceptible population. So these are the initiative that has been taken by the WHO for preventing, yeah, for spreading this diseases. Okay, now don't die because of our ignorance. We have to follow all these things. We have to follow all the routes. And the other thing is that even though if some people have been infected with the HIV, we have to treat them with a sympathy and you cannot shun from the society. Okay, and it's not that if we hug that person who is having the HIV, it will not spread from one person to another. How it is spreading mainly already we discussed, and by the body fluids only it spread from one person to another. So if you are hugging a person who is having a AIDS, it doesn't mean that okay you are also going to get that infection. Okay, and it has to be in a collective manner. The society also has to be involved. Don't treat them, yeah, don't ill treat them. Okay, and it is a melody that this can be tackled by the society, medical fraternity, acting together to prevent the spread of the disease. So again, I'm telling, don't die of the ignorance. We have to see all these things, all the precaution measures we have to take. That's why I took this topic in a elaborate way, so that you should be aware of how this AIDS get transmitted from one person to another, what are the preventive measures that we can use, and how you can educate the other people, or how you can prevent the AIDS from spreading from one person to another. I think all might have understood this topic. If you have any queries related to this topic, you can ask me in the next class. And please go through with your NCERT book. All the details have been given in your NCERT book. With my video, you please take your NCERT book and study this topic. Thank you, everyone.